The limitations in vaccine clinical studies and pharmacovigilance. Initially, I put a disclaimer. Some gene products have been labeled as vaccines. Vaccines, by definition, contain certain amount of known antigen. Meanwhile, mRNA products contain certain gene sequence instead, and this sequence causes the synthesis of unpredictable amount of antigen for unknown duration of time. Few words about me. My background is medical. I was working for 30 years at the Lithuanian University of Health Sciences. I am a pharmacologist. For 10 years, I was deputy director at Lithuanian State Medicines Control Agency, responsible for supervision of clinical trials, for marketing authorization, and pharmacovigilance. I will speak today about bad quality of vaccine trials, blind marketing authorization authorities, pharmacovigilance, so drug safety surveillance, which doesn't work. And I will provide a practical example. How does this work in real life in combination with negligence of physicians during this pandemics of COVID mismanagement? What can I say about vaccine trials? Inappropriate stratification. Short blinded phase. They are not designed to detect rare or delayed adverse drug reactions. They are underpowered critical safety endpoints, and efficacy endpoints were commercial. This is a key table on efficacy. It appears in the summary of product characteristics approved by European Commission, and we go back to stratification. What kind of scientist can make such a stratum? For COVID-19 trial, they put into one group people from 16 to 64 years with totally different risk of disease and complications. People which are completely healthy with those with a lot of concomitant diseases. Next, most important stratum, 75 years and older. People who are most likely to benefit from these products. Which percentage of subjects are here? 4. What? You take healthy people and then extrapolate data for those with cardiovascular diseases, cancer, etc., etc.? Next. Do you see any way individuals with cardiovascular disorders, cancer, immunological disorders, presumably that those were risk groups? No, we have no clinical data. Look at this number. This is subject years, time of follow-up, and this is number of subjects. If you divide this, 6,000 to 20,000, then you get that duration of surveillance was on average 3 to 4 months. They calculated vaccine efficacy. They protected about 800 people of those 20,000 who were included. And they got this 91%. This is so-called relative risk reduction. But absolute risk reduction, so real number of people who benefited from intervention, it is 2, 1, 3% depending on vaccine and depending on age group. But they do not show the absolute risk reduction, violating the guideline of the European Medicines Agency. Conclusion of European Medicines Agency and European Commission was, okay, we can authorize a product on the basis of this data. My conclusion was, first, I should never take this product. Second, marketing authorization authorities have no appropriate qualification, or they are simply captured by pharmaceutical companies. Risk stratification was against science. decision was based on short-term prevention of mild illness. So you prevent mild illness in 3%, let's say, of people, or 2%, and cause at least transient, short adverse reactions, including fever, rash, etc., almost in everybody. I am not interested in mild disease. I am interested in prevention of hospitalization and death. They do not have this data. This did not serve as endpoints of the trial. Then, mass vaccination was done on the basis of conditional marketing authorization, which bypassed full efficacy and safety evaluation. And without any studies on the effect on transmission. The government became most important source of COVID disinformation and misinformation, declaring 100% efficacy against hospitalization and death. This is proof. This is a screenshot from governmental page. Exclusion criteria included pregnancy and lactation, and we see increasing amount of data showing that the products decrease fertility. It might have happened, the similar disaster, as more than 6 decades ago with thalidomide, with tens of thousands of stillbirths and lip abnormalities. We still see those people. They are a little bit more than 60 years old, and you see them with short limbs or even without limbs. So this is Contergan or thalidomide. And now they decided to bypass pregnancy evaluation in pregnancy and breastfeeding. They should have done this because of good clinical practice. They cannot include pregnant and breastfeeding women, but they done even worse. Instead of experiment of a few thousands of women, they have done experiment on billions and billions. It is much worse. Next disaster was a study of Comirnaty, so Pfizer so-called vaccine in adolescents. Without any data on randomized control trials on preventing severe disease and death. Without data on long-term safety, with a short control phase and small sample size. Approximately 1,000 in vaccine group and 1,000 in control group. And without consideration of the number of serious adverse events in the vaccine versus placebo arm. Have a look. 7 serious adverse events in vaccine arm, only 3 in placebo arm. Life-threatening, 2 in vaccine arm, 0 in placebo arm. Of course, this doesn't reach statistical significance. I told already, the trial was underpowered. Publication of me and my colleagues based just on the analysis of the assessment report of the European Medicines Agency. So on my understanding, risk-benefit ratio in this age group is clearly negative. Pharmacovigilance, so drug safety surveillance. I took data from Lithuanian State Medicines Control Agency. They got 35 reports of death after COVID vaccine. Meanwhile, National Data Agency reported 4,962 deaths within 3 months after vaccine. It is 0.7%. What about another adverse drug reactions? I say that percentage reported is definitely less than 1%. Clinical studies demonstrated that almost everybody got some adverse reaction. Now I calculated number of doses administered in various countries and also number of adverse reports I took from websites of competent authorities. So then we get percentage reported, Lithuania 0.18, Norway 0.5, Estonia 0.35, etc., etc. Not so many. So the previous slide may be multiplied at least 100 of times. Health Law Institute encouraged citizens to report suspected serious adverse drug reactions. However, public officials challenged and stopped this initiative. I think that critical drug safety information was lost because of that. Case study: fatal cerebrovenous sinus thrombosis in 17-year-old male, post-vaccine and post-COVID infection. He was misdiagnosed as migraine, received treatment late. Emergency surgery wasn't successful. Laboratory findings demonstrated increased coagulation, extremely high titer of antibodies against receptor binding domain, and elevating troponin, which probably demonstrates cardiac damage. Let's try to follow the criteria of WHO. To assess causality, we need to have a look on temporal link, on biological plausibility, on literature reports, vaccine quality. Temporal link: administration of COVID vaccine community, it's summer of 2021. Then hospitalization due to severe headaches after a few months. After next few months, COVID-19. Mild, only mild illness, and after next 3 weeks, cerebrovenous sinus thrombosis and death. Biological plausibility. S protein, spike protein, it triggers autoimmune reactions against the affected cells, leading to cellular damage. Indeed, there is genetic sequence within lipid nanoparticles, so it may go theoretically into every cell, and then any cell affected expresses S protein on its surface. So this serves as the basis for immune protection, but the same mechanism is responsible for cellular damage. Why? Because the immune system attacks these cells. Once again, this produces immunity, but also this damages the tissue. And consequences depend on the amount of cells damaged and of tissue damaged. S protein may trigger inflammatory cascades, induces the formation of fibrin and fibrinogen resistant to fibrinolysis. So it cannot be dissolved by clotting system, by fibrinolysis. Literature reports. I'm really surprised by the doctors who say, no, this is not possible because this was not reported in the literature. But how did these investigators reported that? If they consider this is not possible, they would never report. Look at Vigilance. Hundreds of reports of cerebrovenous thrombosis and cerebrovenous sinus thrombosis following mRNA vaccination. Vigilance, it is a European database managed by European Medicines Agency, to which reports from all member states have been collected. I think Vigilance lost a lot of reports because of activities of some Lithuanian public officials, which made great damage to public health. Vaccine quality. Maybe it was counterfeit product. More than 10 years ago, European Commission adopted delegated regulation on falsified medicinal products. It was intended to prevent the entry into the legal supply chain of falsified medicinal products. Look what our health minister, famous Dulkys, has done. He made exemption for COVID-19 products. Safety features were not required from December 2020 till at least mid of 2022. So nobody who received so-called COVID vaccine may be sure that he or she received true vaccine. I do not consider process 1, process 2. I simply speak about counterfeit medicinal products. Remember dates of administration to this adolescent? And finally, I make conclusion. The traceability and authenticity of the administered product were not assured. A counterfeit product might have contributed to cerebral venous sinus thrombosis. And I want Lithuanian authorities to provide all evidence to say that I am wrong. Relatives of the deceased applied to the National Service of Accreditation for Healthcare Activities regarding this event. They failed to acknowledge the necessity for probabilistic, not binary analysis for adverse drug reaction. Binary means it caused or it didn't, but it is not possible in most cases. In most cases, you say it is likely, it is probably, it is possible, it is unlikely. But to say yes or no, it is possible very, very seldom. Then, healthcare professionals failed to report the fatal case of thrombosis to State Medicines Control Agency. And neither Lithuanian authority, it is a shame, neither of them until now applied the WHO criteria for the causality analysis of any death following COVID-19 vaccination. Final remarks. I am sure that long-term efficacy and safety endpoints are required in vaccine trials. In most cases, they investigate only immunological response. I am not interested in that. I am interested in how do they protect us against severe disease, against death, and what is the impact of another health parameters. Mandatory review of all fatal and life-threatening events occurring after vaccination should be considered, and independent access to COVID-19 statistics should be ensured. Clinical recommendation. Exclusion of serious pathology should have priority in emergency settings because they misdiagnosed stroke with migraine. Clinicians should be educated on the potential of serious unexpected adverse drug reactions. Unexpected means not appearing in drug label, including the summary of product characteristics. I would like to thank for your attention and to thank all courageous people in Lithuania, Estonia, and all over the world who decided to speak out for the sake of health and human rights. Thanks once again.
Thank you, Rimas. Thank you for the wonderful lecture. And our next speaker is Meike.